Hi, my name is Claire Silkstone, and today I will be explaining the role of variation within the toll interleukin protein domains in plants. Let's start with some background knowledge. The toll interleukin 1 receptor, also known as TIR, is a protein. Part of the protein is the protein domain, which in this case helps with signaling for cell death and with immune system responses in plants. A specific domain that will be talked about in this study is called the BB loop. This study looks at artificial TIR proteins and the variation between them and the BB loops for their effect on signaling cell death and immune response molecules in Arabidopsis thaliana, also known as Thale cress. In this experiment, there were many methods used, but there are three major methods. The first is gene cloning and DNA carrier construction. DNA carrier construction involves taking DNA from one organism and putting it into another organism. As seen in the photo, DNA from the eukaryotic cell is put into a bacterial cell. The purpose of gene cloning in this experiment was to create a large amount of the same DNA from the Thale cress sample. The next method that was used was an immunoblot. An immunoblot is used to look at and characterize proteins, in this case, the TIR proteins. The last method used was consensus determination. This involves the scientists looking at each individual amino acid that makes up one of the TIR proteins. The goal is to find the most common amino acid at a specific location in order to understand the function of each TIR protein. Now on to the results. There are three main results from this study. The first is that the TIR domains of the natural TIR proteins could help design the artificial TIR proteins. The second is that a section of the TIR protein, called the BB loop, in the artificial TIR proteins could help control cell death signaling. And the last result was that variation between the artificial TIR proteins could help with engineering TIR proteins that would either increase or decrease the immune response. The first image correlates to the first result. A shows the process of gene cloning, DNA carrier construction, and the consensus determination. B shows the protein structure of each of the artificial TIR proteins. C and D each show a leaf that has been treated with the different kinds of artificial TIR proteins. E is an immunoblot showing substitutions to the DNA base pairs and how that has affected the TIR proteins. F is a graph that is signaling for the presence of NAD+, which is another protein that works with the TIR proteins to help with signaling. G shows the structure of two proteins also involved in signaling, and when they were active when they were paired with each artificial TIR protein. This second image correlates to the second result. A shows the structure of the BB loop regions in the artificial TIR proteins. B, C, D, G, and G' all show leaves with different artificial proteins. E shows an immunoblot with the regions of the BB loop present or absent and how that has affected the protein. F and H show the two proteins that are related to signaling and when they are active with a specific artificial TIR protein present. The third image correlates to the third result. A is a diagram of a protein in which the domains of that protein will be replaced with artificial TIR proteins. B and E are leaves with the TIRs shown in the specific place to show physical characteristics or phenotype. C and G show the two proteins involved in signaling and when they are active when paired with a specific BB loop variation. D shows the sequence from the artificial and modified BB loops. And F is another immunoblot showing the effect on the protein with each modified BB loop. Image 4 contains a lot of graphs. Graph A plots time and the remaining amount of NAD+ in percentage with each artificial TIR protein. B shows the structure of a specific molecule of ATP, or the molecule that provides energy for cells. C shows the detection of that specific ATP molecule with different artificial TIR variation and BB loop variation. So why does this matter? As the human population increases, so does the amount of food consumed and the amount of food needed. Strengthening plants' immune systems can help them to survive diseases and pests and supply more food for both humans and other animals. A future idea from this study would be to look at the role of TIR protein variation in prokaryotes. Does variation have the same effect in prokaryotes as it does in plants? Here is the article that was explained, and thank you for listening.